Richard Thompson. I'm a professor of biochemistry and molecular biology at the University of Maryland School of Medicine in Baltimore. And we're very interested in new methods for early diagnosis of AMD and also Alzheimer's disease.
I'm Imre Lengyel. I'm from Queen's University, Belfast. And our particular interest is how to translate laboratory observations into the clinic and how to understand what we see clinically, so imaging the back of the eye, and how to match that up with biochemical results.
Dr. Lengyel and I were very interested in zinc in the retina, and most of our work is focused on basically optical sensors, particularly fluorescent sensors of different chemical analytes, metal ions, small molecules, and the like. And Imre was going to come to our lab and do some experiments using retinal specimens, basically, fragments of retinas, and with our zinc sensors. But he stopped off at the synchrotron in Brookhaven. It's not quite a vacation spot, but to analyze some of these drusen, which are deposits in the back of your retina, that if you get too many and too big, they lead to AMD and ultimately blindness. And so he did an experiment with Tony Lanzarotti and Jane Flynn, and just, I guess I could say for the heck of it. And what they found is in the back of the retina, inside these deposits, bone mineral. I mean, like your teeth and your bones. It's a form of calcium phosphate called hydroxyapatite. You couldn't imagine why you would have this mineral, this rock, in the back of your retina. And we knew that people had discovered that the antibiotics, tetracyclines, which probably a lot of people are still familiar with, stick to that mineral. They stain your teeth. But it turns out that it's very, very specific in its binding, and its fluorescence changes. And that change in fluorescence is something that we can measure with a pretty fancy instrument called a laser scanning ophthalmoscope. And that's what sort of got this whole crazy thing on the road. And Imre did some experiments on this that, well, I'll let him talk about them, that basically suggested that this might be the cause of the drusen.
So you think these calcium deposits cause the drusen?
So I think I would rephrase it in the way that calcium deposits are contributing factors to drusen formation and a causal one. So what it means is once this calcification appears in the back of the eye, what it will do is bind proteins, lipids, nucleic acids, and other molecules in the back of the eye, and that's the reason why drusens start growing. So it's one of the components, and we believe that it's a causal component. And why this is important, because we have seen these little calcium spherules, which are really look very pretty, like little pearls on a necklace. But once they are there, they are basically insoluble. That's what we want from our teeth as well. So once they are there, they will be there for a long time, and they will be there to seed any process that is not normal. And what we think is that if the back of the eye is healthy, even if these little calcifications are there, they may not cause too much trouble. But as soon as the cells start becoming ill for whatever reason, then they will start secreting or pumping out material that can bind to these surfaces, and that's what can initiate this process. And what we have been able to show that we have not seen any drusen without some calcification. But as we started to get deeper and deeper into the process, we realized that there is a particular subtype of calcium deposition. We call them nodules, and we showed that clinically these big nodules seem to be associated with a very rapid progression of age-related macular degeneration to end stage. The clinical data showed that within a year there is a 7 times higher chance to develop the final stages of the disease than in those patients without these particles.
Were these calcium deposits found in all eyes or just in eyes that had disease?
So I think just as drusen happens in every one of our eyes, I think we can safely say that it's part of the normal aging process in the back of the eye. But the number and extent of calcification, that is the issue which we need to deal with. Another thing, before I hand it over to Richard to continue how we will use this, is that the Bruch's membrane, which separates the pigment epithelium from the vascular bed, that gets calcified as well. So not just the drusen, which is sitting on the top of this Bruch's membrane, but also the Bruch's membrane itself can become calcified. And these seem to be very early events in the disease process. As I said, they may not develop into disease. So there are other factors, but if this calcification is there either in the Bruch's membrane or in the drusen, then suddenly you have this seed around which the disease starts progressing, and that's what we advocate, that looking at this is critical for an early detection of disease processes.
I mean, there are lots of folks busy trying to make essentially therapeutics that stop AMD, and there are several different approaches that they're using, but we think that we want to be able to differentiate someone who's just, I mean, I have drusen in the back of my eye, my vision is fine right now, but at some point am I going to get AMD? How bad, how soon? It's very common. There's over 10 million people in the U.S. alone who have it, and the question is how soon, how much, and what we're hoping to do is to find a way to measure that by taking a picture of your retina through this ophthalmoscope. And once you know it's there, then maybe you can do something about it. I mean, and like I say, there's a lot of folks who are busy, trying to develop new therapeutics. I mean, this meeting is a very big meeting, but there's lots of posters and talks talking about different sorts of treatments that people are advocating and testing. And what we're talking about, it may not only be useful as a way of finding this out, but also helping to follow the course of the disease and see, is it fast, is it slow, who is it fast, who is it slow for, and maybe relating that to the genetics of the disease, which have been the last 10 years have really gotten our understanding is just leapfrogged. It's just, tremendous in that sense.
You had mentioned that there was a link between macular degeneration and Alzheimer's. Were the same calcium deposits found in both diseases?
Imre's lab discovered this, so the calcification we're talking about in the context of AMD is directly in the back of your eye, in a place called the macula, which is where the disease gets its name. But it turns out that people who have Alzheimer's in what's called the periphery of the retina, which basically is kind of around the edge, right behind where your iris is, you also get calcification. And I would say it's fair to say we don't quite know, how it got there, or what's the, from my standpoint, the molecular process. But recently, again, Imre's lab has shown that you get calcification in the brain as well that's related to Alzheimer's disease. Some of this has been known for a long time, but this is a particular kind of calcification related to a protein called tau, or phosphorylated tau. And that really has got our wheels turning, and we're very actively trying to investigate how that's related to the calcification in the eye and the other pathologies in the brain.
Well, as Richard mentioned, really the interesting fact about the link is drusen deposition and changes in the blood supply, and especially the peripheral retina seem to be affected in Alzheimer's disease. So I think the link is not necessarily between Alzheimer's disease and AMD. The link is between this drusen deposition and the changes in the vasculature, the feeding of the retina through the blood supply, than the actual causes of the disease. Although both genetically and protein-wise, there is a lot of link between the two diseases.
What are your next steps?
Sort of several directions. I think, and from our standpoint, the most important one is we are going to try an animal model of AMD and see if we can see these deposits, in the retina of a living animal, and see how similar they are to the ones we looked at. We looked at an awful lot of these, as Dr. Lengyel mentioned, in the context of cadavers and so forth. Our hope is to come up with an effective technique and something that is going to be, maybe not easy, but certainly straightforward for the clinician to be able to do in his or her office.
So one of the really interesting aspects of calcification, which is sad, we know very little about it everywhere. We know about the bone, we know about the teeth, but the so-called soft tissue calcification is fairly unknown. And we are very lucky. We just recently received a program grant where 4 laboratories with very different expertise will be supported by the NIH for the next 5 years to understand this relationship of how calcification happens, how it would progress. And that's going to be our life for the next 4 or 5 years, because it's hopefully going to pave the way, not just for the detection which Richard talked about, which we think could happen fairly early and could be monitored through this new technology called fluorescent lifetime ophthalmoscopy, but also because we will be able to uncover the molecular mechanisms behind this, and that would allow us to identify pathways which we have not yet explored to stop the progression of the disease.
That's really a key thing. So, we're not only interested in the proximal clinical problem, of identifying people early on and then treating them, but also understanding these processes, down to the molecular level, we think is going to tell us, okay, this is going to be an approach that we can use to stop this. I think that we share a prejudice that calcification is an important part of several diseases, not just AMD, Alzheimer's maybe, but also there's lots of other diseases associated with calcification in terms of hardening of the arteries, things of this sort, and those things are being studied very actively, not in the context so much of the eye, but from our standpoint, the eye is kind of the window to the soul, and it may enable us to understand these calcification processes, which we think contribute to a number of different diseases. And some you can treat, some you can't treat, but if we understand how it works, then maybe we can fix it. And, our vision, if you will, is that we're going to give you a pill to stain the hydroxyapatite, the mineral in your eye, and take a picture that takes about 90 seconds right now, and from that be able to potentially, and again, this is our hope, be able to diagnose Alzheimer's and or AMD beforehand. We're not there yet, but that's what we hope to have.
5 years is the duration of the grant, and we will have several different avenues that we are going to explore, and the timeline is fairly free. The actual ophthalmoscope already exists. It's a commercial device, but not yet in the clinics. So that means that what we need to do now, and that's what Richard and his team is doing, setting up the appropriate labeling method. So one of the big difference compared to the usual devices that are in a clinic is that we will introduce a labeling, a compound that labels the calcification. So we will know exactly not just whether there is something there, but what is there. So that's a significant, gives us significant clarity to what we are looking at. But in terms of when it could get into the clinic, if we are lucky, it could take less than the 5 years. But what will be the next major step after the 5 years grant and the ability to look at these deposits is understanding the molecules behind it, which then opens up new avenues for developing intervention strategies that are very different from those that are in use currently.
Will this research be done primarily at the University of Maryland?
Yes, and we have collaborators at other institutions, Francesca Marassi at the Medical College of Wisconsin, Galia de Bulucina, and I hope she'll forgive me if I mispronounced it. She's at UCSD in San Diego, and at the Sanford Burnham Prebys Institute is Jose Milan, who's just a terrific guy. So we're actually part of a larger team of really talented people. And this has been tons of fun to work with them.
Is there anything you'd like to wrap up with?
Just to say it's been tons of fun, and we were looking forward to more fun. And the harder I work, the more fun I have. So and I think that's probably true for Dr. Lengyel as well.
Yeah, I think it's really exciting to work on this interface of that is patient-facing as well as laboratory explorations. So it is, let's say, ping-pong between looking at what happens in a patient's eye and then looking at what happens with the molecules in the back of the eye, and then exchange that information is the one which is we call translational ophthalmology, which is, I think, the most exciting part of ophthalmic research these days.
Thank you both for sitting down with me. We wish you much success in your new project. We look forward to hearing from you again.